Our dream situation would be if we can lower the levels of AGEs, that you can actually have your cake and eat it too, and not get the problems that we normally associated with hyperglycemia.
humanOS. Learn, master, achieve.
Hello, everyone. Welcome back. Today, I'd like to welcome Pankaj Kapahi to humanOS Radio. Pankaj is a professor at the Buck Institute, and for those who might not be familiar with it, the Buck Institute is an independent biomedical research institute that just so happens to be in my backyard in Marin County, California, that is devoted solely to research on aging. Pankaj has a very impressive research background as he discovered the role of something called TOR, or target of rapamycin, in mediating the effects of dietary restriction on health and longevity. More recently, his lab has been working on how molecular clocks and fat metabolism influence the effects of dietary restriction. But he and his team are also working on advanced glycation end products, or AGEs. Now, what are AGEs? Well, if you've ever browned meat or baked bread, you've seen the formation of them in action. And unfortunately, they are delicious, so our palate nudges us to consume them, but they are likely an underappreciated contributor to the aging process. So first, Pankaj, welcome to humanOS Radio. It's great to have you with us.
Thank you very much, Dan. Great to be here.
What exactly are advanced glycation end products?
So every cell that utilizes glucose splits glucose into 3 carbon atom molecules. And unfortunately, not all metabolic reactions are clean in the body, which means when one product is being made to another product, there are some byproducts that are made. So one of the byproducts of utilizing glucose is methyl glyoxal, and there are other reactive carbonyls that are made. And these byproducts increase the formation of advanced glycation end products. So the idea is that they are reactive, and then they will bind amino acids like lysines and arginines, and then they will modify them. These reactions are irreversible, which means they will accumulate in the body, and there isn't an easy way to get rid of them. And that's the problem with advanced glycation end products, and their buildup is damaging in several diseases.
How were they discovered?
Very old literature by Monnier and Cerami, where they identify the reaction that is called the Maillard reaction. The science is almost more than 100 years old, and they discovered the effects of what we come to know as glycooxidative stress, modification of glycating agents on amino acids.
It was hypothesized decades ago that AGEs might play a role in the aging process. Why has it been so difficult to make a causal link between AGEs and age-related disease in humans?
I read about advanced glycation end products when I was a teenager, and I got very excited about this. And you're right, it's not had the traction I think it deserves. And one of the primary reasons I think is hard to quantify the buildup of these products. I mean, the way I look at it is aging is less of biology and more of chemistry, in the sense that biology is a program that's there to help us develop and maintain health. But with age, when things fall apart, that's more like the process of rusting. That's where molecules like advanced glycation end products and those processes become critical and are underappreciated because they're very heterogeneous. So there are many types of reactive carbonyls that modify amino acids, DNA, and lipids. They're hard to measure, and they have not really been studied in what I'd call more genetically tractable models. So there are over 10,000 papers showing the existence of this and their accumulation with age. But whether any one of them or some of them are causal has been harder to understand.
Where does the Maillard reaction occur in the body?
It's happening both in the mitochondria and outside. The unfortunate thing is some of these reactive molecules like methyl glyoxal can actually go across membranes. There is a number of enzymes that have evolved to deal with them. They're in the mitochondria, but also in the nucleus and in every tissue. So there are defenses in place in multiple tissues and multiple compartments to make sure that you're not accumulating these reactive dicarbonyls, which make advanced glycation end products.
They're occurring in the body all the time, and we have then a detoxification systems for them. What is the major pathway that AGEs are detoxified?
Multiple glyoxylases now being discovered. So I think it almost argues for the importance of reactive carbonyls. It argues if there's so many enzymes in place, then there must have been a role for them. And that's what we're finding, that if you mutate one, then you build up significantly. What's even more interesting is if you knock two of them out, then effects are additive. So it argues that multiple redundant enzymatic defenses are in place to keep these glyoxylases at bay. This is only the first part of the story. After they modify macromolecules like lipids, amino acids, or proteins, they can also be removed after that. And that happens more through normal protein turnover. AGEs are then found in our serum, cerebrospinal fluid, and they're excreted through the urine. So it's especially interesting under situations of kidney disease. So in chronic kidney disease, there's actually a buildup of advanced glycation end products. And the idea is that it causes inflammation. So this is a positive feedback. If the kidney is already damaged, it's not going to clear advanced glycation end products, which is going to further enhance the inflammation by the buildup of AGEs. Those are some of the things that are known. I think there may be other defenses that we don't know about, but those are the two main things we know about in terms of removal of AGEs. One by dealing with the precursor of AGEs, and secondly, by excreting the fully formed advanced glycation end products.
There's late and early stage Maillard products. The earlier steps of age formation may be reversible. The later ones, once age is fully formed, they're just hard to break down. Can both be a target for interventions that could help to either keep them at bay or to reduce them once they've formed?
A very interesting clinical trial was done for stopping the accumulation. It was an age breaker. So the idea was it would stop the later product from forming. But unfortunately, such antidotes have harmful effects. Because you can imagine if there's something that's reactive that is trying to block the formation of AGEs, the antidote might itself be toxic. Unfortunately, that clinical trial, even though it had great data from animal studies, in humans, there were some side effects and that had to be stopped. So I think that's another reason that perhaps the field of advanced glycation end products got stalled. But to your question, I think there might be more to gain by stopping the precursors of AGEs from forming. And that's how the body seems to do it. The approach we're using in the lab is letting the organism tell us how to deal with this best. When you use the power of genetics, you can let the organism tell you if there's more stress due to, let's say, glucose or buildup of these reactive dicarbonyls, which would cause AGEs, how would a cell deal with it? And we're learning lots of new things from cells this way.
Is there any physiological function for AGEs? Previously, we thought reactive oxygen species were simply a byproduct that was harmful, but we found out that they actually have a physiological role as well. Has a physiological role for AGEs been identified, or is it simply at this point just a problematic side effect of biology and chemistry?
The history of methyl glycol is over 100 years old as well. It was initially ignored by biochemists because this is a metabolic byproduct. It's not a reaction for which there is an enzyme. So initially, biochemists focused on the reaction where there was an enzyme. And I think that some of these metabolic byproducts might be relevant for aging. So that's why we studied them. There might be a signaling role for AGEs. Like, for example, when you have increased glycolysis, you want to shut down utilizing fat. There are certain situations where you want to use high glucose, for example, under states of high growth. So we think they definitely regulate physiology, and under certain situations that might be beneficial.
In the beginning, I alluded to that AGEs are found in another source. So we know that they're produced endogenously, but we also know that they're produced in our diet. How can people reduce AGEs in the food that they're eating? Are there specific foods or cooking techniques that should be avoided?
Since I've gotten into this research, I've changed my lifestyle a little bit. So I am not popular at parties because every time there's barbecued food in front of me, I can't help telling people that this creates the formation of AGEs that are detrimental to us even when we consume them. So we are making AGEs endogenously, but we are also getting it from our diet and also from our microbiome, in fact. Any cell that uses glucose actually makes AGEs. So bacteria are also making AGEs. So how do you reduce the intake of AGEs? Avoid cooking techniques that involve dry heat, the oven or using barbecue, or wherever there's charring and browning. Those are things to avoid. What I've started doing more is steaming and boiling foods when you need to cook.
A great product to be invented would be something that would allow us to cook in more a gentle manner by lower heat that doesn't produce the AGEs, but could somehow replicate the flavor of these AGEs and the crispiness that is done through frying and barbecuing. Any food scientists out there, let's work on that.
Why is it tasty for us? This is another question we talk about at lab meetings and we don't have an answer. But interestingly, we found that even invertebrates find the AGEs tasty.
We've been cooking with fire for hundreds of thousands of years. That's probably why we have multiple pathways that are dedicated to help handle these. But our environment has changed so rapidly. We might just be consuming foods that are outside of the window of what the body can handle successfully. Talk about why these are problematic. I understand that they accumulate. We can go into the RAGE pathway, which is the receptor for AGE. How does this contribute to chronic inflammation?
Yeah, so one of the deleterious side effects of having advanced glycation end products is that it enhances inflammation. And one of the key receptors that has been defined for this is the activation of RAGE by the binding of an AGE, a molecule to RAGE. It argued that AGEs are not just building up and inert, but they can play a signaling role and they can activate inflammation. So it gave a useful therapeutic target. And inhibiting RAGEs pharmacologically or genetically has been shown to be protective. However, what we are learning in the lab is that AGEs bind many other molecules, and RAGE is just one of the receptors. So that's the exciting work we're getting into to find other targets for AGEs, which might explain lots of other behaviors, like why we eat more, why it affects obesity, why it causes neurodegeneration, and why it causes inflammation.
Once AGEs bind to the receptor, there's a signaling cascade that will increase nuclear factor kappa beta, oxidative stress, inflammation. These are all things that we know associate with chronic disease. One disease that comes to mind has been known for a long time is the association with diabetes and microvascular complications. What is this connection between AGEs and vascular health?
The strongest evidence for AGEs has come from the link between diabetic complications and AGEs. If you are in a state of hyperglycemia, then you're more likely to build up the precursors for AGEs. That would then elevate the levels of AGEs systemically. If you do that for long enough, you're prone to several diabetic complications. We're not talking about diabetes per se, which is simply defined as hyperglycemia. We're saying that if you have sustained hyperglycemia over decades, then you increase the risk for several disorders. These include retinopathy, nephropathy, a risk for chronic kidney disease. More than half the patients who have chronic kidney disease have diabetes. It's one of the leading contributors to also cardiovascular diseases, increased risk of stroke. And also we're learning recently that diabetics have 2 to 4-fold the risk for Parkinson's and Alzheimer's as well. So if you have diabetes, there's an increased risk for several diseases, which we think are all part of the diabetic complications. So one of our ideas is that it's not glucose that's causing these diseases. The most damaging product that you're making in diabetes might be the downstream advanced glycation end products. They are the ones that are contributing to these multiple diseases simultaneously.
Is it particularly hyperglycemic states that are problematic, or can AGEs be formed under normal glucose diet wrapped in whole foods? Traditional cultures, some had very high carbohydrate diets, and they don't seem to have as much chronic disease. Now they have a very large phytochemical load that comes with the natural food in natural form. So can some of the effects of AGE formation be offset by certain phytochemicals or other factors that are part of the diet? And do you have an entirely different situation where you have, let's say, the same percentage carbohydrates in your diet, but it's in a whole different form of refined foods, low phytochemical load? What's your thinking around there?
So the load of advanced glycation end products is going to depend on multiple factors. So this includes the consumption of AGEs from the diet. So as has been noted, just the increased consumption of more cooked food can actually increase the risk of diabetes. So that's definitely one contributor. The second part is the amount of glucose, carbohydrates they are taking, mainly a simple glucose molecule, that will increase glycolysis. You will build up AGEs at a faster rate. And the third part of the equation, for which we need more understanding in terms of precision medicine, is different genetic factors that are important in the detoxification of AGEs. So, as you said, someone could be eating normal amounts of glucose but still build up AGEs. And that's already been known that AGEs build up also under normal aging. But the question is, in certain situations, it's not going to be toxic. It's not going to cross this threshold where it becomes pathological. But in certain people, they may not even have diabetes and they'll start seeing some of the deleterious effect of AGEs. So we need to understand the genetics better to understand who are the people who are more at risk, either through dietary consumption, through intake of carbohydrates, or just the genetic factors that are important for detoxification.
So this is definitely a moving target here. So there's genetic susceptibility, there's diet and percentage of macronutrients, possibly time of life. You could plausibly argue that having a lower carbohydrate diet throughout life might help to offset aging as well by reducing AGEs.
That's true. So lower carbohydrate diet has been shown to extend lifespan. And one of the factors that you definitely see improvement on is reducing advanced glycation end products. Definitely, I think it's one of the contributors. And I think now more people are paying attention to the effects of ages on multiple systemic effects.
You mentioned the connection between neurodegenerative diseases. What is the evidence for AGEs and some of these conditions beyond what you've mentioned so far? Let's start with Parkinson's disease.
Parkinson's disease is perhaps one of the best examples because we know about 7 key genes that have been associated with early onset Parkinson's disease. And one of those genes is called DJ1. And that gene turns out to be a glyoxylase. So it's one of those enzymes we describe like a glyoxylase whose function would be to detoxify the precursors of ages. So we're finding, for example, the loss of that builds up the advanced glycation end products and leads to neurodegeneration. Just like that, the evidence is building up because now when you look at patients with Alzheimer's or Parkinson's disease, postmortem brains of people with neurodegenerative diseases has shown that there's a significant buildup of AGEs in their brains.
Plaques extracted from Alzheimer's disease brains show 3-fold increase in AGE content versus age-matched healthy. They seem to be accumulating in the plaques in Alzheimer's. Is this a line that is being investigated carefully now as well? The connection between AGEs and Alzheimer's disease?
That's true. That's one of the projects that's going on in our lab. If you can reduce the buildup of AGEs, does that protect against pathogenesis in Alzheimer's disease? And the basis of a number of diseases may be common. The idea is that AGEs are enhancing the vulnerability of proteins to aggregate. If you're seeing more cellular stress, and if you see more protein aggregates, that might hasten the process in some of these neurodegenerative diseases.
If we're thinking about protein aggregates, it makes me think about fasting. That might be something that is beneficial to clearing out protein aggregates in the body. Has there been a connection or any fasting research that you've seen that has identified a reduction in AGEs, or is that something that has not yet been looked at?
Some of the most dramatic effects we have seen in our lab on reducing ages has been under fasting. Even if you take a genetic mutant that is more prone to building advanced glycation end products. So this is an animal that's missing the glyoxylase. So this can make almost 1,000-fold more precursors of the advanced glycation end products. This animal fasted shows the lifespan is almost no different from the control animal. The effect of fasting can be that dramatic in these animals. There's a lot of research arguing that fasting can reverse the effects of diabetes. It definitely applies here because it would work at many levels, from the production of AGEs to the detoxification of the precursor of AGEs and to the clearance of the more mature forms of AGEs.
That's great news. I just saw a paper yesterday, but 2 years of fasting completely reversed diabetes. Not only did it have an improvement in HbA1c and other markers of diabetes, but they actually were able to tolerate carbohydrates again. So ARCs are part of the pathway to detoxify. So NAD, pH-dependent aldo-keto reductases. There's great interest in NAD plus stimulators, NMN or NR. These are supplements that you can take now. David Sinclair is doing a lot of work with that, others as well. Could supplementation with NAD plus assist ARCs in detoxifying AGEs as we get older?
Yes, definitely. That's a very interesting idea. And I think we're exploring a number of commonly used drugs and supplements in our lab to see which ones influence advanced glycation end products. Nicotinamide is potentially one of them.
One paper that I found particularly exciting was this one that examined the synergistic impact of combined polyphenol therapies, so hesperidin and resveratrol, in improving AGE outcomes. So one induced glyoxylase 1 and also had an impact on activating NRF2, both of which can seem to detoxify and remove AGEs. Is there any additional work that you are aware of looking at natural phytochemicals to improve AGE situation?
A number of researchers are working on this. I think the paper you mentioned is one of the recent studies, but I think there are a lot of active researchers in this area.
It's always a little frustrating to me. We live in a world where if you can't capitalize on it, it tends to get underfunded. And yet it's nice to see when work on these compounds that reinforce a healthy lifestyle, things that are available to us in the grocery store right now. Reducing the amount of AGEs that you consume seems like a smart approach. But if you're eating anything like barbecue, making sure that you're getting with some phytochemicals. You could even just start with hesperidin and resveratrol. So that's going to be like lemons on your food with a small glass red wine maybe, and that might handle it better than if you were to eat the barbecue with a beer.
Eating the rain was always good advice. In salad and greens on the side, I think that's the best thing you can do. Yeah.
AGEs are talked about less often in the public sphere. And through this excellent review that you just did, which I learned a ton from, are championing that this needs to be part of the conversation. What do you think are the next critical questions to address in order to advance the field a step farther?
This area is ripe for a lot of discovery. One of the things I think we'll see a lot more with the help of genetics is how diabetics become vulnerable to different diabetic complications based on your genetics. So, for example, imagine you come in with diabetes, but it could be predicted early on that you're more likely to have peripheral neuropathy or retinopathy based on your genetics. Having this sort of information could be really game-changing because through understanding how AGEs affect several diseases, we could have a better lifestyle approaches for people who have diabetes or at more risk for particular disorders. Understanding the genetics of that and how the buildup of AGEs causes pathologies in different tissues is definitely one area that's going to be very exciting. The other area that you already touched upon, the therapeutics, I think we should be able to see a lot more therapeutics in this area because as we're learning from the genetics of simple invertebrate organisms, they also build up advanced glycation end products and they also have the same enzyme systems in place to deal with them. So we'll be able to use the power of genetics in these model organisms to find better therapeutics which would be relevant for mammal. And we already have a few in our lab that we are actively exploring. These are some of the areas where we could do a lot more, both in terms of understanding the disease process and coming up with better therapeutics, which specifically deal with AGEs as a target. We're also learning a lot more about AGEs and its role in cancer and inflammation and many other diseases. As that gets built up, the case for understanding the genetics for how AGEs are built up and the therapeutics will just get stronger and stronger.
You mentioned how fasting was useful. What was the fasting regimen in that study?
We've fasted worms, C. elegans, that we use for our research, which build up AGEs. We found that they build up almost 1,000-fold more AGEs. And when we fast them, we found that they don't show the pathology that we see in a normal mutant animal. And the pathologies we found that were rescued by fasting include neurodegeneration and lifespan. And the fasting regime that we used, we've done complete starvation in these. But we've also done dietary restriction, which in the case of C. elegans, we limited the food by a quarter to a tenth of what it normally eats, and that can rescue the pathological effects.
But that was more chronic than having them, let's say, fast once a month or periodically, whatever the time scale was.
Correct. We haven't done all the different forms of fasting. Also, one thing to add was adding glucose to the media exacerbated the effect. So glucose, again, is a key player here. Simple sugars. So having a more complex carbohydrate diet, reducing your glycemic index might be one of the key things here to lower the production of AGEs.
Is it a certain temperature that we want to stay below for cooking?
I don't have as much knowledge on this, but what I do know is as you increase the temperature, the number of chemical species you end up generating is significantly higher. And this goes beyond AGEs. So the lower you can stay in terms of the heat you use, the better it is. The slow cooking would also be beneficial than a higher heat cooking. I wanted to add one more idea. Please. One of the key hypotheses we are testing through our work on advanced glycation end products is that it's not glucose that is the key molecule that causes pathology in diabetes, but it's the buildup of the downstream products like AGEs that are the real target. Of course, lowering glucose helps in diabetes and will help reduce AGEs. But if we understand the real culprits, then we can come up with better therapeutics to both deal with specific diabetic pathologies and lower the mortality risk due to diabetes.
You could imagine a scenario where somebody has a diet that does contain carbohydrates, but has other factors that's a part of it that is limiting the propagators that turn into these end products that are problematic. That could be a fruitful area for intervention. If you can cut them off, then you're going to also limit the late stage implementation of AGEs into your cellular structures. So on one hand, the earlier forms cause inflammation. The late stage AGEs are harder to get rid of. There is some biotechnology techniques getting into it. But in the meantime, cutting off the source would be a good way to go about it.
Our dream situation would be that if we can lower the levels of AGEs, that you can actually have your cake and eat it too, and actually not get the problems that we normally associated with hyperglycemia.
Pankaj, thank you so much for your time and your work here. It's opened my eyes to a lot of things. We have a diet as part of something on HumanOS called the Flash Diet and Simple Food Diet. And we've been recommending lighter cooking, stewing, slow cooking techniques for a long time with the recognition that AGs do contribute to some of these pathologies. But my knowledge here deepened a lot with your review. So thank you for aggregating all this information into this excellent review for the different projects you've got going on.
Thank you very much for having me.
Thanks for listening, and come visit us soon at humanOS.me.